Hello, everyone. Thank you so much for joining us at a special look of what our restoration teams are doing on a ship in the deep Gulf. My name is Erin Spencer. I am the communications manager for the mesophotic and deep benthic communities restoration work, and I am going to be your host today. Today, we're going to get to talk to some friends that are on the NOAA ship Nancy Foster, a 186-foot vessel that is currently based in Charleston, South Carolina. Right now, the ship is located about 121 miles from Gulfport, Mississippi. Today is actually the last full day of operations that they have on this particular leg of the mission. So we're super excited to be able to talk about some of the work that they've done over the last couple weeks. So throughout today's program, please go ahead and share your questions in the chat and we'll pull those up and share it with our folks on board. And we will pause for questions as we go, but feel free to put them in anytime. Also, at the end, we would love to get your feedback from today's program. So please fill out this survey to let us know a little bit about your experience before you log off today. We'll send this link now, but we'll also send it at the end of the program. So anytime. Okay, so in just a couple of minutes, we're going to connect with the folks on board and we're going to talk about what they've been up to and what life is like on board. But first, we're going to talk a little bit about what they're doing out there and what this restoration work means. As you travel deeper in the ocean, less sunlight is able to penetrate the water. Just past recreational scuba diving limits, you reach the mesophotic zone, which means middle light. Despite the low levels of sunlight, this zone is home to colorful corals, sponges, fish, and invertebrates. Beyond the twilight zone, there is no sunlight at all, and the temperatures get colder and the pressure gets higher. But there are still vibrant habitats full of life at these depths, which are hundreds to thousands of feet below the surface. These mesophotic and deep benthic, or seafloor ecosystems, are found worldwide, including in the Gulf. And they're full of this rich biodiversity, including some of these great creatures that we have captured on previous missions. However, in 2010, the Deepwater Horizon oil spill dealt a heavy blow to the Gulf communities on land and at sea. 11 people lost their lives, and natural resources, wildlife, and habitats across the northern Gulf were severely impacted. More than 770 square miles of deep sea habitat were injured by the spill. But as a result, collaborative restoration projects are currently underway to help us understand and restore these seafloor ecosystems below 160 feet. The projects are developing new and improved maps of the seafloor, learning more about the species that live in these habitats, the environmental conditions they thrive in, and how they're connected across the Gulf. Determining the best methods to encourage deep sea coral growth and reproduction to restore impacted coral species. Reducing the threats that these ecosystems face, including marine debris. And connecting the habitats and restoration work to the public and natural resource managers through events like the one we're doing today. This restoration work is made possible by the Deepwater Horizon Natural Resource Damage Assessment Open Ocean Trustees, and is led by the National Oceanic and Atmospheric Administration and the Department of the Interior. Plus, many partners, including a large number of academic institutions and non-governmental organizations. And you can visit this link here and scan the QR code to learn more. Okay, now that we have a little bit of background about some of this restoration work, I think it's time that we hop to our friends on this NOAA ship Nancy Foster so they could tell us a little bit more about what they've been up to. Hi, here we have Jayla and we have Eric. How are you guys doing?
Good. How you doing, Aaron?
Doing great. Thank you so much for joining us today. We're really, really excited to hear what you've been up to. Do you want to first introduce yourselves and tell us a little bit about what your role is on board?
Hi, everybody. My name is Jayla Jones, and I'm an aquarist at the Mississippi Aquarium, and I'm an aquarist trainee on board here today.
And I'm Eric Cortez. I'm a professor at Temple University in Philadelphia, and I've been working on the ecology of the Gulf communities for a few decades. And I'm the science lead out here working on both the coral propagation work and the habitat assessment.
Amazing. Thank you both. And I think let's get started just by talking a little bit about this mission overall. Can you tell us what some of the goals are that you're trying to accomplish while on board?
Yeah, there's two really main goals. One of them, like I said, is the habitat assessment, and then some of the coral propagation, which I'll let Jayla explain. But the habitat assessment, we're really going back to a lot of the places that we've been visiting since the spill happened, and even prior to that, to look at the health of the corals, to understand where the corals live and the conditions that they live under, so that we can better protect them and it can inform some of the other projects that are going on.
For coral propagation techniques, we are doing live coral sampling, and we get our sample, we bring it on board, and we are essentially trying to see what they like, what they don't like, what they like to eat. Can we get them to spawn and potentially outplant them back into the Gulf for restoration?
That's awesome. And we definitely will have some questions about that and what it means to keep the corals alive and thriving. And actually, I want to point out we are looking at some footage now from the remotely operated vehicle that is currently underwater as we speak, and doing some of these transects, looking at some of these sites. And while we're looking at this, you mentioned collecting some samples. And I know one of the goals here is to look at the diversity, genetics, and reproduction of these corals of interest. Can you tell us a little bit about why we need to understand the population connectivity and genetics of these corals, and essentially how does that help in restoration?
Yeah, I mean the population genetics, what that means is seeing how related corals in one place are to another, corals of the same species. And as Jayla was saying, if we're going to outplant these corals, if we're going to bring them back into the environment, we want to make sure they're coming from the same population, so that we understand what kind of habitat they like, and we can put them back in a place where we can restore a degraded habitat, but we know that they're going to survive.
Excellent. And when we talk about outplanting, just for the folks at home to understand, can you tell us what you mean by outplanting?
So for outplanting, after we have potential spawning in our lab, we have what we call recruits, or our little babies. And so we have those just growing on tiles. And once they are healthy enough and large enough to be outplanted, we will have them essentially brought into the ship, and we will have them,
The biobox.
or the bio box, yes, but I was talking about more of a frame that we
Oh
would
yeah,
place down in a set for reef habitat.
yeah.
Hmm.
We're working on a bunch of different kinds. This is the one that you're seeing now is from the mesophotic sites, where they're bringing a bunch of the corals back in their nice cold water to bring them back down and outplant them, and that's being done a lot of times with technical diving, which is pretty amazing, or by ROV. And there's some of the racks, frag racks.
Yeah,
That's
a
what they
better
are.
term.
Thank you. That JLo was talking about. Deeper down, we're really just they're way ahead of us in the mesophotic areas. We're still experimenting with this down deeper.
Hmm.
When you get below the mesophotic zone, when you're deeper in that twilight zone at the depths that we're looking at right now in this ROV feed, it's a little harder to keep the corals alive. They're really, really sensitive to temperature. If they're really deep down, we can't really keep them in the aquarium on land for very long, because they require the higher pressures down there to basically keep them intact. We think we don't fully understand that. So some of the corals you're looking at now we can keep in the aquarium, so we're experimenting with them now. I've had them in my aquarium in the past in my lab and done some experiments to find temperature tolerance, or some their resistance to other types of variables in the environment. The ones deeper down, we're bringing up, and then we're fragmenting them into smaller pieces that will grow faster, and we're mounting them on these substrates and putting them right back down so that they are only on the surface for this minimal amount of time, and we keep them nice and cold. And the bio box that I was referring to is this nice like insulated box that, the surface waters of the Gulf can be 90 degrees Fahrenheit, but down there it's like 35 degrees. And so if they heat up too much, they won't survive. So we have to be really careful about that.
And I think you touch on something that's really important about this type of restoration work, which is a lot of what we are doing in the Gulf with these corals is pretty new, right? Like working with these mesophotic corals and deep corals and having them in captivity. We're just learning so many new things because it really is a lot of work that hasn't been done at this scale. Um, I want to come back a little bit more in a second to talk about what it's actually like keeping them in tanks. But thinking about, all of these different objectives that you're trying to accomplish on this mission, can you talk to us about the planning that goes into, the weeks and months leading up to something like this? What's involved in trying to have a successful mission?
Yeah, months and years in a lot of cases. I mean, these things are planned well in advance. This MDBC program is really amazing because we have so many opportunities to get out to sea. Um, and we're working in so many different kinds of environments. You need to figure out what your objectives are, figure out what kind of ship and what kind of ROV or the technical divers can accomplish those objectives, and then put your team together. And this is actually one of the things that I like the most. You have to find the right people that can work together out here in really close quarters for a long period of time and have the right expertise to get everything done that needs to get done on board. So you're looking at Jason, our ROV pilot right now. Marina is one of my students. Randy was over there. We got people logging data and taking notes and, paying attention to everything that we're doing and all the samples. And we do that on every single one of these cruises, recording all the video entirely so we can go back through it later. And just, packing and making sure you have the equipment you need. And do you want to talk about the aquaria that are out here?
Absolutely. I was going to say beforehand with deep water corals, because the tanks have to be so cold, cold water bacteria takes a lot longer to cycle than a warm water bacteria tank. So we have to cycle our tank for about 2 months beforehand. So just dropping your temperature in your tank and making sure your bio does not die, essentially, and adding more bacteria if you need to keep the tank going. Dosing ammonia every week, making sure it goes through the nitrogen cycle, testing your water quality parameters. And so once we get the tank cycled, then that's when we can confirm to people who are out here sampling, like, it's okay for us to bring corals back in the lab, because if not, there's no way to keep them, essentially. There's just not proper parameters to put them in. So we want to put them in the best environment possible. We want to match everything environmental wise that we see in the benthic region that we are sampling in. So we'll take some of the data that we have here and we'll try to match our tank extremely close to what the conditions are to keep the corals happy as possible.
Gotta take care of our babies.
Yes.
Exactly. And Jayla, you and I were talking about this a little bit before we started, but just really in awe at the level of care and detail that's required, and all of this expertise that you have as an aquarist keeping these animals alive. And right, it really is the center of all this restoration work, and we need to do everything we can to keep them healthy and thriving. You mentioned a couple of these, but can you talk us through, when we're thinking about the critical conditions and care that's required to keep these corals alive, what are the things that you are checking and looking at in the tank when you have corals in the tank?
So the first main thing that I'm checking for is phosphate levels, ammonia levels, and alkalinity levels. We want to check ammonia levels because any kind of ammonia in the tank is bad. It is completely off limits. If you have ammonia in your tank, the best thing to do is to do a water change and try to get it out as soon as possible. Same thing with kind of high nitrates, going through the nitrogen cycle. If your nitrates get too high, same thing, do a water change, try to get it cleaned out, keep everything in lower. I would say around, for phosphates, point 0.1 would be the highest because they really don't like high phosphates. And ammonia, 0.01, honestly, they really don't like ammonia. And then for alkalinity, anything between 7.5 DKH to about, I would say, 10 DKH is where they're happiest. But just to keep all of those parameters in check, I do different types of water quality tests a day. So I'll do an ammonia test, or I'll pull out my white side. I'll make sure I check the temperature, and I'll make sure I check the pH in the tank as well. So just making sure I keep my levels balanced. The best thing for the corals is stability. They appreciate a stable tank, for sure.
I also appreciate a stable tank. I feel like I can relate to that with the corals.
Yeah.
You're keeping them alive, not just when you take them back to the aquarium on land. Are
Mhm.
there parts of keeping those corals alive at sea that are more complicated when you're doing it from the vessel?
Absolutely. When we bring the corals up and we have to process them, they are not allowed to touch air at all. So we have to put the quiver into the cooler. We have to make sure we have a good amount of room in the cooler to pull them out. And so when in the video, Matt right here, we're moving the corals around with this forceps. We wear our gloves. We try not to touch them. Handling causes a lot of mucus buildup, so we try to keep that as low as possible, honestly, because the more mucus they develop, the more you have to water change the tank. And so in these tiny coolers, we have dividers or egg crates or plexiglass as well. And we just like to keep the corals from touching. So it makes it a little harder to process because you're limited on space.
That totally makes sense. And there's folks in the chat, they're really enjoying this and just saying, yeah, you need to try to replicate conditions of the ocean as much as possible to keep everything stable. As a reminder to those folks joining in, you can add your questions in the chat or anything that you want to chat about with our scientists here. And we're keeping the corals alive, right, is one element of what's going on the vessel. And as we were talking about some of the other goals, and it's also tuning back into thinking about this ROV transects that we're doing. Eric, I'm interested to hear some of the other samples that you're collecting on this expedition. What are some of the other types of samples that you're collecting, and what are the things that you look for when you're looking to take a sample, whether that's one of the other biological samples or an actual coral sample?
Yeah, there are so many different things. I mean, the physical samples that we're taking, we take water samples sometimes to analyze the water chemistry to find out some of those parameters that JLo was just explaining. But we'll take coral samples for reproduction. We're trying to figure out when they reproduce and how they reproduce. And a lot of them, corals don't have just one way of reproducing. They can reproduce asexually, they can change sexes, they spawn at different times. There's so many different things, and that they're all different. Um, so we're trying to figure that out so that the aquarists, when they have them in the lab, they know when to look for a spawning event. So
Mm-hmm.
you got to turn off the filtration. You got to do all this other stuff to make sure that you collect the tiny little babies there that are being produced in the lab for putting them back out in the environment. The population genetics that we talked about, or you have like the relationship between these different populations. Sometimes we're trying to figure out what a species is, and that sounds very simple and sounds like something scientists should know, but it's something that we actually struggle with a lot. I'll give you an example. In the deeper benthic sites, at almost a mile down at one of our sites, there was a species that was impacted. You actually saw it on the intro video, that had the oil and dispersant on it when we first found it. Almost the anniversary was 2 days ago, actually, 16 years ago. And we came out when we found those corals and they were impacted. We, like, visually, when you look at them, it looks like they're all the same species. And by doing some of the genetics work, it turns out that there's actually two different species, and we've now learned how to tell them apart, but it's difficult. So, if you're managing a species, if you're trying to figure out how many you've lost and how many you need to replace, you first have to know what that species is. So it's really, I mean, going all the way back to really basic stuff like that. This species that you're looking at right here, we understand a lot about that because people all over the North Atlantic and South Atlantic have been studying this one for a while. Its traditional name was Lophelia, which is what we still call it, but it's Desmophyllum pertusum. Um, Another example, it was just re-described, and it turns out it's more closely related to another species of coral that only forms these little polyps. And so we thought it was closely related to something else. It turns out from the genetics, it's related to this other one, and we had to change the name. So it's really, a lot of fundamental stuff, but you have to understand that in order to understand how the population works, how the species works, and what we can do to intervene and restore some of these communities.
That's amazing and a really perfect example of how much there is to still understand about these habitats and, just how much we have learned even in the last 16 years from the spill, especially about these habitats in the Gulf. And that actually kind of relates to one of the questions that came in from the chat. Someone is interested to know what would be your reaction if you find a new shark species, but I'll expand that to even include these invertebrates as well, right? When you're finding a new species, what would be your reaction?
Oh my gosh, somebody take a picture. Because we want it documented for sure. If you don't get it on picture, there's no proof. Just kidding. But no, I would definitely take a picture and
Oh, I think we might have lost our stream. Can you hear us okay on board?
can you
Yeah,
hear me now?
Okay,
yeah, we
great.
can hear
Yeah,
you.
you
Okay.
guys are
Yeah.
back. So
Okay.
we lost you at, oh my gosh, take a picture, right? We want to document everything.
Okay, but yeah, so other than that, other than taking picture and video, we I tried to personally ID it as or try to get close to genus as possible on board. um I've been flipping through ID guides all week long, so that's my first step as somebody who is very interested in taxonomy.
I mean, there's a lot. There's a lot of corals. A lot of them are difficult to tell apart, and people don't really realize that there are deep sea corals, cold water corals. In the Gulf, we have like 100 different coral species, and we're trying to tell them all apart and figure out who's related to who and where they live and how they're doing after the spill and monitor their populations. It's just telling them apart is a challenge.
And I think it's also a great time to talk about, there's a lot of this field work that's happening, right? But this is really just the beginning or a continuation of the work. All those samples when they come, when the cruise is done, the expedition is finished, they all go to different areas for further analysis. And so when you're talking about, finding new species, a lot of that comes, as you said, they're really hard to identify visually, and so a lot of that information comes down the road as they've gone to different partners and for analysis. And so a lot of those eureka moments are in other labs across the country as opposed to just while you're on board. But yeah, I think it sounds like it would be very exciting.
Yeah, we
Um,
have some great partners at the Smithsonian Institution in Washington D.C. They're, trained taxonomists. They describe species. They can tell them apart. They see the differences in them that we can't see out here. That they take a microscope and the genetics and all that and put it together. And it's a great team over there.
that's fantastic. We have another question from the chat from Chris. What is the most unexpected thing that you've seen on this expedition so far?
Hmm.
Well, I'll do
Yeah, go
one
ahead.
while you think about it. There's a site that there's the Mississippi Canyon, which isn't far away. It's a little west of where we are now, and a site that I visited a few times a while back, about 20 years ago. Um, And we returned there and we explored a different part of it that we hadn't been to. And we found just the biggest coral colonies that I'd seen at that site, or really of this one species almost anywhere in the Gulf. And it's just amazing that, we went there over and over again. We've been going for 20 years and you can still just, oh, well, let's go see what's on that spot, and it turns out to be the best thing at the site. They're really magnificent, and it's really fun. It's really rewarding, and it really, again, reminds you like how much more exploration needs to be done.
Yeah, they were absolutely huge. I think my unexpected thing would be, because I'm in the dark room of the aquarium all the time, being on board and just seeing the corals in general has been unexpected for me. But seeing different species and different colorations has also been super exciting for me. Um, I have really enjoyed just seeing the different colors of the corals, even though they're the same species that I work with back at home.
It must be also such a wild um, kind of experience to be working with them on land and in the lab and be so like close up, and then to see them again, in the wild on the seafloor. It's such a powerful experience. It's also partially why we really enjoy events like this and are so appreciative of your time here, because it's an experience that very few people get to have, right? Seeing those corals up close, and it's just so powerful. I also, on that kind of note, I want to kind of take a broader look back and ask you both about this career path in particular, because a lot of people are curious, right? As I said, not a lot of folks get to be up close with these corals. Can you tell us a little bit what initially drew you to this type of work?
Well, I'll start because mine's probably going to be a little shorter. But
Are you calling me old?
no, 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 no. But I started out. My dad actually works for NASA, so I spent a lot of time as a kid looking at the stars. So once I got past looking at the stars, I was like, okay, Dad, I want to do something else. Like I want to go look at the ocean. And so my first time on the beach as a kid, I'm just hoarding all the seashells in a Bratz purse container. But that's kind of where I started. And then after that, in high school, I went to a career fair and I started talking to a career scout down at University of Southern Mississippi in Hattiesburg. And I went there to get my degree in marine biology. I talked to my dad a lot about it actually because I was also interested in engineering. And he told me to follow my passion and to follow my heart, even though, as an engineer himself, he was like, engineering is very cool. So, but after that, I worked really hard on my undergraduate. I got into an undergraduate lab by myself. I worked really hard to do that. I wrote a lot of letters. I talked to Dr. Nicole Phillips a lot. So after I worked in her lab, I moved on and got some temporary jobs. And then found this job because when I was about 9 or 10, the oil spill happened. And I'm from Alabama, even though I'm from North Alabama. It did have an impact. We used to go to Orange Beach all the time. And just seeing the beach completely different changed my whole life, honestly. And so now that I worked with the corals, it's been exciting. I started as an intern for a couple of months, and then I moved to part-time position, and then I was part-time for about 7 to 8 months, and now I'm the full-time position. It's been a lot of fun. I've learned so much. I've never been in Aquarius before, so this has been a journey of a lifetime for sure.
That's amazing. Also, what an incredible arc from the stars to the deep sea, right? That's so special.
Thank you.
I'll try and keep mine short, but my dad also worked for NASA for a little
Really?
while.
Wow.
Yeah. Long time ago on the Apollo missions.
Wow, that's cool.
Not for very long, but a little while. And yeah,
Cool. Very cool.
Yeah, I know. I didn't know we had that connection. And that is inspiring. Just that kind of exploration and that following your passion and going to really achieve your goals. I was the same, playing on the Gulf Coast as a little kid and going fishing with my grandfather. I grew up outside of Boston, but my grandfather lived in Gulfport, Mississippi, so we were down here a lot. Um, and a place up in New Hampshire called Star Island that we used to go to. And by the time I was in second grade, I knew this is what I wanted to do. But it took me a while to figure out what being a marine biologist meant. Uh, I went to undergrad in Long Island, and then I took a few years off. I wasn't sure if I wanted to go to grad school, and then I got a master's degree at Moss Landing Marine Labs in California. A master's degree that took me 6 years while I was working full time, and I ended up teaching at community college, and then getting written into some research grants, and then finally now I'm teaching and doing research, and now I figured out that maybe it's academia, but by then I was almost 30 years old. So it took me a little while. So then I got my PhD, and that was when I started really working in the Gulf of Mexico again. And the Gulf, really has been a part of my story the whole time. And it's been really amazing to spend so much time working on these communities in the deep sea that I didn't even know existed when I was playing in the bayou and on the beach,
Right.
in southern Mississippi.
And you kind of alluded to this, but you've been involved in Gulf in deep coral work in the Gulf, including in the early days after the spill. Can you talk a little bit about what it's like to return to some of these places over and over and kind of see the change over the last 16 years?
Yeah, so my first research cruise in the Gulf was actually 2001. So it's it's hard to
More
believe,
than
but
16 years?
it fit 25 years. And we've been visiting a lot of these sites. The site that we're at today actually is one that we have been visiting for. Others have been here and visiting for over 30 years, and have been able to watch it change. Things change in the deep sea very slowly, um, but we are seeing things like fishing line at this site. And yesterday we were on a site where we saw these big weights that were used for deep drop fishing that's happening more and more in the Gulf. And we did surveys on a lot of the oil platforms. And in 2009, we were on a research cruise, and we went right past the Deepwater Horizon, and didn't even really think about it. It's just another rig out there. So when this happened in 2010, it was really, kind of a shock. And it was everybody just mobilized and tried to get out there, tried to see what was happening. Um, we honestly, when we were talking to each other, we thought oil floated and the deep water communities might be okay. But it turns out that through a lot of different processes, a lot of it came raining down or snowing down as marine snow, came down as marine snow and affected a lot of these places. And seeing that, when we saw those first images, it was really sad. And we immediately knew that this was something that we were going to be doing for a while. But it's really amazing that working with NOAA and this program, that we've gone from that damage assessment process that happened then, where we were really just like gathering evidence and working with lawyers. And we've gone from that to, after the settlement, the money from the settlement has to go back into restoration of the habitats and the organisms that were impacted during the spill. So it's been really rewarding to be able to do that, to be able to kind of give back and try and protect these places and try and actually directly restore some of them. You alluded to this before, Aaron, but this hasn't really been done anywhere, at least definitely not on this scale. So we're working with, we're talking with a lot of other people who are also pioneering some of these techniques in Spain, in California, in Norway, and Sweden, in the UK, and we're working with them and exchanging ideas. But the stuff that we're trying here is really cutting edge, and we're figuring it out as we go along. And we've started at kind of a small scale to make sure some of this stuff is going to work, but pretty soon the techniques that are working, that are effective, we're going to scale them up and really put these communities back together.
I love that you brought that up, Eric, because it's so important, right? We're talking about this restoration work being so collaborative, and it really is. It brings together so many partners, but the work is not limited to the Gulf. It's happening in the Gulf, but the impacts of this restoration work and what we're learning about how to care for these corals, and I mean that's going to have impacts all around the world. And so, just thank you again both so much for the work that you are doing out there, and thank you for sharing some of your time with us today, especially in such a busy expedition that you're on. I want to just end with one additional question, and it's great because actually Chris in the chat has something really similar. But what is it about this work that makes you optimistic, right? That gives you hope about the restoration?
I think I would say seeing my corals extremely happy after I worked so hard on their tanks. Um, it is probably the most rewarding thing to see, having them spawn and being able to have recruits in your lab. It is almost like a pat on the back for all of your hard work. Corals don't really do anything fast, okay? They do everything in slow motion. So to see even the smallest amount of basal growth, or oh my gosh, he's decided to develop another branch on, on his axis. I'm so excited. So just going through and seeing those little victories really does make it worth it, for sure.
Yeah, the stuff that the Aquarius are doing is really amazing, and they've really perfected it at the mesophotic level, and now they're working more in the deep water species. I'm really excited and optimistic that they're going to be able to do the same thing. That we're raising new corals in the lab and putting them back out in the environment. That's incredible, and just to see. Just the fact that there are so many people working on this, that so many people really care about it, it's bringing the deep sea to a lot of people. And Aaron, I want to thank you and all the communications team for doing that, because that's, people need to hear the story, and they need to know that these things exist, and they need to know about the good work that's being done here, because this is an optimistic story. This is, in the end, it's a success story, and people all around the world are watching what we're doing and learning from it, and they're going to be able to do a lot of these same things when we have other human impacts in other areas around the planet.
Well, I appreciate that shout out so much, and appreciate both of your work, Jayla and Eric, and your time today. It really is such a incredible thing to be able to share this. And so thank you to everyone who joined as well. With that, we can all say, I'll say on behalf of everyone joining us, goodbye and good luck for your last full day of operations. And we are wishing you a very safe and very smooth and not rocky ride back to port tomorrow. So thank you both so much. Have a great rest of the expedition.
We'll see you soon.
Oh yeah,
Yeah.
see you later.
Very soon. Oh,
Bye.
Thanks.
I wanted to
wait, one
answer
more
this question.
Oh, go for it.
I do watch anime. I've been watching One Piece the whole time, and this is my Cowboy Bebop case. I hope you really enjoyed that.
I love that. Oh my gosh, thank you so much.
Of course.
All right, we will talk to you both soon. And thank you to everyone in the chat for adding your questions, and for everyone joining us today. And today during this program, we really wanted to focus on this one element of the mesophotic and deep benthic communities restoration work that's going on. But this is really just one expedition in a suite of lots of work that's going on all year long. It's just really a snapshot of some of the projects in action. But and just a reminder, these projects involve this really extensive mapping on the seafloor, diving deep to understand the seafloor communities and how they're connected. Really, and we heard about this today, this groundbreaking coral propagation work that's happening in the lab and at sea. And then other things like removal of marine debris and other threats. And then also just, again, connecting folks at home to the habitats that are in the Gulf that are just so special and so important for the health and for the Gulf and those who depend on it. If you enjoyed the program today, don't forget you can check out some of our other live streams. We have another one coming up on May 6th that is all around how we're using tech diving for restoration. And so please join us for that. The information we'll share in the chat, a link that you can find not just the upcoming live stream, but actually all of the previous live streams that we've done. We've covered a lot of different topics, including going in depth with ROV pilots and what life is like at sea. We've talked to all kinds of folks. So please be sure to tune into some of those. And then there's a couple other links that we're going to share in the chat for other resources about the MDBC portfolio and projects, some educational materials that are free and ready to use in classrooms and at home. And then again, last but not least, we would love to get your feedback on today's program. We're going to share a link in the chat for a little feedback form. Please feel free to share anything that you really enjoyed, anything that you would like to see in the future. And yeah, we are just so excited that you joined us. And thanks again.